Hey everybody, it's Dr. Gilchrist, and I am here to talk to you about a brief introduction and overview of the nervous system. This is going to correspond with Chapter 2 in your textbook. Now, one of the things that I've mentioned in previous lectures is that drugs don't work by magic. For you to understand drugs, you have to understand a little bit about how the nervous system works as well. And part of the reason for this is how drugs ultimately end up working. They work on the mechanisms of different parts of the nervous system, including receptors and neurotransmitters, and thus they are able to affect and create different changes in the brain, and thus those changes to things like our mood, our perception, our cognition, and our behavior. So let's go ahead and give a brief overview of what we're going to be talking about today. So we're going to start by talking about the different cells in the nervous system. And then we're going to talk about the major components and divisions of the nervous system as well, including spending a little bit of time talking about cerebrospinal fluid and a little bit of time talking about genes. Then we'll finish up by talking about brain injury and why it can be so difficult to restore the brain to its prior state. We'll kind of talk about some of the difficulties with that. So we're going to start by talking about the different cells of the nervous system. We can basically break down the nervous system into two major cell types. So first, we have the neurons, which are functioning as our structural building block of the nervous system. Their job is to transmit information not only to other neurons, but to things like muscles, glands, and organs through both electrical signaling and, as we'll talk about in the next lecture, chemical signaling as well. And that's when our neurotransmitters come into play. We'll then talk about the glial cells as well. So glial cells, interestingly enough, you actually have more glial cells than you do neurons. You have about 10 times as many glial cells as you do neurons, despite the fact that we call this the nervous system. They are there to provide support for neurons. We'll talk a little bit more about them later, but their primary functions involve insulating neurons and potentially protecting them from damage, providing nutrition to neurons, and also providing them with structure. So now let's continue to talk about neurons. So here is an image of a neuron. You can see it right in front of you. Neurons are specialized animal cells that are designed for communication. So we're going to start by talking about the soma. The soma is basically located right here. It is also sometimes known as the cell body, and it contains all of the different components that you would find in any other cell in your body. It contains the nucleus with chromosomes and DNA, and it does include the other organelles. Then we have our dendrites. Dendrites are designed to receive incoming information from other neurons. And then we have the axon, which you can see right here. The axon is basically there to transmit information. It's basically going to conduct an electrical signal all the way down to the terminal buttons. And ultimately, these terminal buttons contain neurotransmitters, and they're going to release those neurotransmitters into what is called a synapse so that information can be communicated to other types of neurons. Now, neurons can ultimately come in 3 different varieties that I am sharing with you here. So we have what are called motor neurons, we have our sensory neurons, and we have what are referred to as interneurons. Sensory and motor neurons, as you can see here, have axons that actually project pretty far away from the cell body. So as you can kind of see, the job of the motor neuron is to basically send information from the brain to the muscles so that the muscles of the different parts of our body can move. Sensory neurons are going to take information from sensory receptors throughout our body, like our eyes, our ears, and our skin, and they're going to send that information through a sensory neuron so that this sensory information can be sent to the brain. Motor information comes from the brain, and so for this reason, sometimes motor neurons are referred to as afferent neurons. Sensory neurons are sending information to the brain, and thus they are sometimes referred to as efferent neurons. Now, we also have interneurons. Interneurons are cells that are entirely contained within a given structure. So interneurons may only be found in the brain or entirely in the spinal cord. They do not branch out to muscles, and they do not make contact with sensory receptors. They are located entirely within the brain and spinal cord, and their job is to communicate with other neurons. So how do neurons ultimately receive information from other neurons? So as you can see in this image, if you take a look at neurons on a slide or in a picture, if they're very closely packed together, it may actually look like those neurons are touching, but in fact, they are not. There's a very small gap that occurs between neurons that is referred to as a synapse. So information from other neurons is received and sent across this small gap. Now, these gaps can be located between the axon of one neuron and the dendrites of another neuron, and this is referred to as an axodendritic synapse. It begins in an axon of one neuron and it terminates on the dendrites of another. We can also have what is called an axosomatic connection. The connection begins in the axon of one neuron and terminates on the soma of another. Axodendritic synapses are the most common. Axosomatic are the second most common. And you have a variety of different receptors that are located on the dendrites and the soma of neurons. So once those neurotransmitters cross into the synapse, they will make contact with a receptor site. And thus, the cell that is receiving this information will either be excited, meaning it's more likely to fire, or it's going to be inhibited, meaning that it's less likely to fire. We'll talk a little bit more about what these excitatory and inhibitory messages do in our next lecture on neurotransmission. Now, the soma, as I mentioned, contains chromosomes and it contains DNA. The nucleus ultimately contains chromosomes, which are strands of DNA, and they also contain genes. So genes are sections of chromosomes that code for very specific proteins. Your cell nucleus contains about 46 different chromosomes. And in addition to the different components, in addition to chromosomes, we also have polymorphisms as well, and transcription. So polymorphisms are kind of the different ways that chromosomes may exist. There may be different types of alleles or different types of mutations. But chromosomes also contain transcription factors, which will either increase or decrease gene transcription. Now, I mentioned that genes code for proteins. These proteins will only be coded and turned on in certain types of environments. As a result, a very interesting field that has arisen is a field known as epigenetics. Epigenetics basically refers to the study of mechanisms of how genes express themselves that do not involve directly altering DNA sequences. We see how the environment that we grow up in potentially changes how these proteins are coded. Now, I want to go back to talking about those glial cells that I mentioned at the very beginning when we talked about the building blocks of the nervous system. As I mentioned, glial cells are various types of cells that provide support to the neurons in a variety of different ways. So glial cells are often going to play a supportive role. So we have cells like oligodendrocytes. They help myelinate and provide a fatty substance with axons to speed up. They wrap these fatty sections around axons to basically speed up transmission. So going back to our picture of our neuron, you can see here that the axon is coated with myelin. Myelin is a fatty substance that ultimately speeds up transmission within a neuron. And it's the oligodendrocytes that do this. Oligodendrocytes are present in the central nervous system, and they wrap around many axons at once. Schwann cells do a very similar thing for the peripheral nervous system, but they only myelinate one axon at a time. Astrocytes are star-shaped cells that provide structural support. They also maintain the balance of ions in the cell. They provide nutrients, and as we're going to talk about a little bit later, they contribute to the formation of something called the blood-brain barrier. The blood-brain barrier is a protective function for our neurons, and we'll talk a little bit more about that as we move into talking about different types of drugs and talking about things like pharmacodynamics and pharmacokinetics. The blood-brain barrier prevents certain types of drugs from actually affecting the brain. So for a psychoactive drug to work, it needs to be able to cross this blood-brain barrier. The astrocytes, as we'll talk about at the end of this chapter, also provide a response to injury when neurons are damaged. And then we also have cells like microglia. They remove waste, and they also provide an immune response because your central nervous system does not have an immune system. There are no helper B cells and T cells. So the microglia are basically going to provide that immune response. So now let's talk about the different components of our nervous system. So there are two major divisions of the nervous system which are mapped for you here. The central nervous system is marked in peach. The peripheral nervous system is marked in lavender. The central nervous system is basically made up of the brain and the spinal cord, as you can see right here. And you know that it's the central nervous system because this is protected by bones. Then we also have our peripheral nervous system, which is made up of the nerves that branch out from the brain and the spinal cord. We can further divide the peripheral nervous system into different subsections. So we have what your textbook calls the somatic nervous system. It is also sometimes referred to as the skeletal nervous system. And then we have the autonomic nervous system, which can further be subdivided into the sympathetic nervous system and the parasympathetic nervous system. So here's kind of another way of looking at these different divisions. So the central nervous system is made up of the brain and spinal cord. This is where sensory and motor information is integrated and it's basically analyzed. We have our somatic system, which is made up of the spinal and cranial nerves. That includes our sensory nerves that respond to different things in our environment, and then the motor nerves, which help our body to move. The autonomic nervous system is made up of the sympathetic and parasympathetic branches. Now, one thing that I'll show you in a few slides is that the sympathetic nervous system and the parasympathetic nervous system are completely kept in separate sections of spinal cord. The sympathetic nervous system is basically made up of nerves that branch out from the thoracic and the lumbar spine, and then the parasympathetic system is made up of the cranial nerves that branch out from the back of the brain and the sacral section of spinal cord. And they're basically there to provide energy or to conserve energy in our internal environment, including areas like the smooth muscles for things like digestion, our cardiac muscle, and our glands and our organs. So let's start by talking about the peripheral nervous system, and then we'll move into talking about the central nervous system. So the skeletal or somatic nervous system is made up of the spinal and the cranial nerves that take information from our senses or help our body to move. And so the spinal nerves in particular carry both sensory and motor information. They are separate from each other, but they are located within the same sections of they are located within the same portion of the spinal cord. So sensory neurons, our afferent fibers, carry information from our senses to our brain and our spinal cord, and they are located in the dorsal horn of the spinal cord. Now, in the spinal cord, the dorsal horn is going to be near our back. Then we have our motor neurons. Our motor nerves are efferent fibers. They carry information from our brain and our spinal cord, and they are located in the ventral horn of spinal cord. That's going to be located on the front of our bodies near our stomach or facing our stomach. So here's kind of what that looks like. So here's our dorsal horn, here's our ventral horn. So here's the dorsal horn, this is going to be located near the back. Here's the ventral horn located at the front of our body near our stomach. You can actually see the sensory neurons terminate here in the dorsal horn, and you can also see that our motor horn terminates in the ventral portion. Despite all of this, despite the fact that they're segregated into these horns, notice that the fibers are mixed in these portions of spinal cord. Now, what about the cranial nerves? Now, we have 12 pairs. I'm not going to go into a lot of detail about what these do. These cranial nerves will include things like the optic nerve from our eye, our auditory nerve from our ear, also some motor nerves and some mixed nerves that are both sensory and motor in nature. So we have 12 pairs of cranial nerves that largely serve sensory and motor information in our head, our face, and our neck. Now, most of these nerves are going to be mixed, but some of them aren't. And in those cases, you may have a sensory-only nerve, such as the optic nerve, or you may have a motor-only nerve, such as the oculomotor nerve, which moves the eye. Now, some of these cranial nerves are a part of the sympathetic nervous system if they are involved with glands and organs. For example, the 10th cranial nerve is what is referred to as the vagus nerve. It is a part of the parasympathetic nervous system, and it branches out to a lot of other glands and organs. Now, let's move on to talking about the autonomic nervous system. So the job of the autonomic nervous system is to basically regulate automatic functions of our nervous system. This will include things like our heart rate, our respiration, our digestion, and our body temperature. And it basically provides contact with glands and innervates and makes contact and connections with the smooth muscles of organs. And as I mentioned before, we can divide these into two different divisions, the sympathetic branch and the parasympathetic branch. So the sympathetic nervous system is sometimes referred to as our fight or flight system. This is going to be activated during periods of physical or psychological stress or when we're exerting ourselves. And generally, we're going to find that the goal of the sympathetic nervous system is to provide the body with energy to fight or run from challenges. So you can see here that our sympathetic nervous system is located entirely within the thoracic portion of the spinal cord and the lumbar portion of the spinal cord. Notice too that in some cases, these nerves do not contact glands directly. So you can kind of see that there are some that do, but there are some that are not. So we have areas of connection between preganglionic fibers, which use acetylcholine, and postganglionic fibers, which use the primary neurotransmitter of the sympathetic nervous system, norepinephrine. But you can see different actions that the sympathetic nervous system will take. When you're stressed, your pupils will dilate. Salivation is a form of digestion. It will be inhibited. The airways will relax. Heart rate will increase. Sweat glands will release sweat. The liver will stimulate glucose production and release. Glucose is our body's biggest source of energy. Digestion will be inhibited. The blood vessels will constrict. Epinephrine and norepinephrine will be released by the adrenal glands. The bladder will relax, and ejaculation will be stimulated in people who are assigned male at birth. Now, the parasympathetic nervous system does opposing actions to the sympathetic nervous system. The purpose of this system is to conserve energy and recover when stress is over. So we're going to see opposing actions to the sympathetic nervous system. And whereas the primary neurotransmitter of the sympathetic nervous system is norepinephrine, the primary neurotransmitter of the parasympathetic system is acetylcholine. So you can see here that the parasympathetic nerves are located entirely within the cranial nerves and the sacral portion of the spinal cord. The pupil will constrict. Salivation will be stimulated, digestion will be stimulated, the heart rate will slow, respiration will slow down because the airways are constricted. The gallbladder will release bile to basically metabolize glucose. The blood vessels in the intestines will dilate for further digestion. The blood vessels will dilate in the skin. The bladder will contract. And in the case of the sex organs, we will get erection and clitoral engorgement being basically a product of the parasympathetic system. So as I mentioned, each of these two divisions has its own primary neurotransmitter. In the sympathetic nervous system, it's a little bit more complicated. Axons that come into contact with the target tissues are going to release norepinephrine, but prior to those preganglionic neurons that are not making contact with those target tissues, they're actually going to release acetylcholine instead. But the ones that come into target tissues, those are going to be the more critical ones. Norepinephrine is critical here. For the parasympathetic nervous system, postganglionic neurons and those target sites are going to release acetylcholine. So it is kind of interesting that acetylcholine is involved in both of these divisions. Now let's finish up by talking about the central nervous system. Now, I mentioned that the central nervous system is largely protected by bones, but we also have the meninges, and as we'll talk about in the next slide, cerebrospinal fluid. So we have three different layers of the meninges to help protect the central nervous system. We have the outermost layer, the dura mater, which is very tough, and it is called the tough mother for a reason. Just below the dura mater, we have the arachnoid layer. This is a web-like sublayer called the subarachnoid space that is filled with cerebrospinal fluid. And then finally, just on the outside of the brain, we have the pia mater, or the soft mother, and this comprises the thin innermost layer of the meninges. So as I mentioned, the cerebrospinal fluid provides additional protection. So it surrounds both the brain and the spinal cord. In the brain, you can find that in those 4 ventricles, and you will also find it in what is called the central channel of the spinal cord. Now, in addition to providing cushioning and protection, cerebrospinal fluid also aids the exchange of nutrients and waste between the blood and the brain. Cerebrospinal fluid is formed in an area called the choroid plexus. You have a choroid plexus in every one of your ventricles, and this is an area where we actually do not have a blood-brain barrier. And because of that, we can actually do that exchange and filter out cerebrospinal fluid. Now, there are 6 different regions of the central nervous system. Your textbook is not really going to talk about this as much as I will, but I wanted to share this with you. So you can kind of see this in a developing brain. You can see those 6 different divisions. So initially, the central nervous system basically begins as a neural tube. So you can kind of see here that at 25 days of embryonic development, we basically have just a tube. By 40 days, it is beginning to differentiate into something that looks more like the human brain that we are used to. And we can actually see the beginnings of areas like the forebrain, the midbrain, and the hindbrain, as well as the spinal cord. 10 days later, we have developed, the forebrain has further developed into an area called the telencephalon, which basically makes up the cerebral cortex, and the subcortical structures that make up the diencephalon. By 100 days, you can see the cerebellum, the pons, the medulla, and the spinal cord. So over time, we are beginning to see that differentiation. So here you can see kind of the major structures, the forebrain, the midbrain, and the hindbrain. And we can further break the forebrain up into the telencephalon, which is largely made up of the cerebral cortex and a few subcortical structures. So we have the neocortex, the basal ganglia, and the limbic system. And then we have the diencephalon, which is made up of the thalamus and the hypothalamus. We have the mesencephalon, better known as the midbrain. Then we have the metencephalon, made up of the cerebellum and the pons right here. We have the myelencephalon, which is the medulla, and then that branches off to form the spinal cord. Let's talk about each of these divisions in a little bit more detail. So here's kind of another way to look at it. So here we have a ventral portion of the brain. You can see portions of the frontal lobe. You can see the temporal lobes right here. Here we can see a view of the cerebellum. We have our midbrain located right here, our pons, and our medulla. And then you'll know that you're looking at a ventral portion of the brain by noticing a couple of different things. First of all, you can see the pituitary gland. Second of all, you can see the optic chiasm, where the 2 optic nerves from each retina of the eye cross over. And then you can also see the olfactory bulbs, which are our sensory organs for smell, kind of sticking up just under the frontal lobe. Here is a mid-sagittal view, so we're basically cutting the brain from the center. We'll know that we're looking at a sagittal view of the brain because we can see the corpus callosum that connects the 2 different hemispheres. Here you can see areas like portions of the limbic system, including areas like the fornix, the cingulate gyrus. You can see the pineal gland, which is important for sleep-wake behavior. Again, here's the pituitary. Here is the thalamus and the hypothalamus. You can see the colliculi of the midbrain. You can see the cerebellum, and again, the breakdown of the midbrain, the pons, and the medulla into the spinal cord. So the spinal cord is made up of cell bodies and myelinated axons. So what's kind of interesting is that when we talk about the brain, gray matter tends to be on the outside. Myelinated axons, that white matter, tend to be on the interior of the brain. This is reversed in the spinal cord. So we can actually see the gray matter, the cell bodies, are in the inside of the spinal cord, and the outside of the spinal cord is our white matter, our myelinated axons. You can see the coverings of the vertebrae of the spinal column as well as the meninges. Here's our dura mater, our arachnoid space, and our pia mater. And we can see the spinal nerves, both the motor and the sensory nerves, branching out from that spinal cord. It is going to enlarge into the brainstem at the point that it reaches the skull. So going into the brainstem or the myelencephalon, this is going to include areas like the medulla, and the medulla is going to be critical for a lot of life-sustaining functions, such as things like heart rate and for respiration, as well as vomiting. So in particular, one area that you might be interested to learn about in the medulla is something called the area postrema. It is sometimes referred to as the vomiting center. And the vomiting center, we have an area where the blood-brain barrier is reduced. And you might kind of be wondering, why would we need a reduced blood-brain barrier for this vomiting center? Well, on the off chance that you were poisoned, or you take drugs that are potentially detrimental, things like alcohol poisoning. That reduced blood-brain barrier is going to allow those types of drugs or poisons to come into contact with this area, allowing vomiting to happen and potentially working to get that drug out of your system. Moving on up, we have the pons and the cerebellum, which make up the metencephalon. A few critical areas that we're going to talk about later on throughout this class. First of all, one area is the reticular formation. This is going to be really critical for things like arousal, our state of alertness, sleep-wake behavior, muscle tone, as well as attention. But I want to talk about two other critical areas in the metencephalon that are going to be really important. And that's because these areas are involved in neurotransmitter production, or they involve neurotransmitters. So first, we have an area called the locus coeruleus. This is a nucleus that sends axons to the forebrain, areas like the cerebral cortex. This is the source of neurons that use norepinephrine. So this is where norepinephrine neurons originate. We also have what are called the Raphe nuclei. This is the source of neurons that use serotonin. So here we are talking about major neurotransmitter players. So when you hear about the locus coeruleus, think about norepinephrine. When you hear about the Raphe nuclei, think about serotonin. This area is going to be, these neurons are going to influence things like sleep, aggression, impulsiveness, and mood, and generally serotonergic neurons that originate from the Raphe nuclei are going to be inhibitory in nature. Additionally, we have the cerebellum. The cerebellum is a sensory motor center. It takes information from our senses and helps us with our motor movements to navigate varieties of different environments. So the cerebellum receives a variety of different inputs from our senses, as well as information about our current position in space, where other people are in space, and how our balance is currently doing. And the reason for that is to help us navigate different sensory environments. And it's going to coordinate this sensory information with the motor information that it receives. Moving on up to the midbrain. The midbrain is made up of the tectum and the tegmentum. The tectum, we're not really going to talk about as much. The tectum has some visual and some auditory centers here, but they're not really important for this class. On the other hand, the tegmentum has a few different areas that are going to be kind of critical when we talk about different types of neurotransmitters and different types of drugs. So first of all, we have the periaqueductal gray. Now, if you had me for biopsych this past spring, you might remember the periaqueductal gray from discussions of both predatory and defensive aggression. In addition to these things, the periaqueductal gray is a pain center and has a very large concentration of opioid receptors there. Opioids, by their nature, are pain relievers. So the periaqueductal gray is going to be kind of important when we talk about the opioids. Next, we have the substantia nigra. Axons in this area go to the basal ganglia, and they're very large users of dopamine. Dopamine is going to be critical for a lot of motor behaviors and a lot of habit learning behaviors that the basal ganglia engages in. We also have an area called the ventral tegmental area. This is also an area where we find a lot of dopaminergic neurons. And these axons are going to go to the limbic system. And for those of you who had biopsych, you might remember me talking about the mesolimbic pathway. That's what this is. And also, those axons project to the cortex to form the mesocortical pathway. So here, just to kind of show you the basal ganglia, here's our substantia nigra. And then that's going to project to areas of the basal ganglia, including the caudate, the globus pallidus, and the putamen. So now let's talk about the diencephalon. We have the thalamus and the hypothalamus. So the thalamus is basically our body's sensory relay center. Basically, all of our senses, except for smell, go to the thalamus before they are processed in their respective areas of cortex. We also have the hypothalamus. This helps our body maintain a balanced internal state. And for the purposes of this class, it also controls the autonomic nervous system and it controls the endocrine system, our glands that are involved in the release of hormones. Now let's talk about the telencephalon, which is made up of the basal ganglia, which I just showed you, the limbic system, and the cortex. So the basal ganglia, technically, if you've had me for biopsych, you know that it should probably be called the basal nuclei because we are in the central nervous system. But this area is critical for things like movement and habit formation. We also have the limbic system, which is made up of the cingulate cortex, the fornix, the mammillary bodies, which are located right here, the hippocampus, and the amygdala. And the limbic system is going to be really critical for a lot of our emotional responses, motivation, learning, and memory. And the limbic system, particularly the mesolimbic pathway, can be hijacked, and thus we process a lot of reward there, and drug addiction may happen by hijacking that mesolimbic circuit. Now, a little bit more about the limbic system. We have areas like the hippocampus, which I've shown you right here. This is an area that's really critical for memory formation and for spatial memory. In addition to this, we have also found that high stress can damage cells in the hippocampus. And we also know from research that people who are prone to depression have a reduced volume of hippocampal cells. So it can potentially be used as a biological marker for depression, and it is often a subject of treatment for that as well. And then we have the amygdala, which is going to be really critical for those emotional responses. It's very implicated in processing emotional stimuli. Then finally, we have the cerebral cortex. The cerebral cortex is made up of gyri, which are raised portions of cortex, and deep grooves that are known as sulci. A deeper sulcus like this one, the lateral fissure, well, is called a fissure. So we're going to see a lot of fissures, sulci, and gyri. The cerebral cortex is made up of 6 layers of cortical cells, and as I mentioned, gray matter is on the outside, white matter is on the inside. So if you actually looked at the cerebral cortex, it would appear gray because of the larger number of cell bodies that are contained within it. Now, each hemisphere has 4 lobes: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. Going back, the frontal lobe is here in blue, the parietal lobe is here in orange, the occipital lobe is here in purple and pink, and the temporal lobe is here in green. Each of those hemispheres has a primary cortex, which is for providing conscious awareness of sensory stimuli, and in the case of initial processing. An exception to this is the motor cortex. The motor cortex is the primary motor cortex is the last part of the brain that processes motor information before it's sent to the spinal cord and the muscles. For the parietal lobe, we have the primary somatosensory cortex. For the occipital lobe, we have the primary visual cortex. And for the temporal lobe, we have the primary auditory cortex. Now, there may be a secondary cortex, and these and other association areas help process higher order information, more complicated information. They also integrate information from other parts of the brain and help lay down multisensory memories. Now, in psychopharmacology, as your textbook is going to talk about, rats are often going to be the research models. Now, there are going to be some similarities. Notice that the rats tend to have a very similar structure in their major locations of the brain. And that's because, like us, rats are mammals. However, notice that we have considerably more association areas than rats do. We also have more grooves, we have more fissures and sulci, and that's to basically help increase the surface area of the brain. Here's just another different view of that. So here's our motor cortex, our somatosensory cortex, our auditory cortex, and the visual cortex. Notice that in the rat, somatosensory and motor have been combined into one area. The visual cortex is here, the auditory cortex is here, and there's very few other areas for those higher order processes. Now, locations in the brain are going to be based on a neuraxis. This is an imaginary line that starts at the spinal cord and then heads to the brain. This ultimately helps describe locations in 3 different dimensions. So here we have anterior, here is superior, here is posterior, and here is inferior. Now, because our brain is rotated 90 degrees from the spinal cord, at least in the spinal cord, nothing here really changes. So that part's really nice. When we get into dorsal and ventral, it's going to get a bit more complicated because these are going to be rotated 90 degrees. Dorsal is the top of our brain. Rostral is toward the nose. Caudal is toward the back. Ventral is down. This is all going to be rotated by 90 degrees in the spinal cord. Medial is the inner surface or an inner view of the brain. Lateral is the outer surface of the brain or of the spinal cord. Additionally, we can talk about different planes of section in the brain. So here we have a horizontal plane. We basically cut horizontally through the brain. A coronal cut sits directly on top of the brain like a crown and goes straight down. And then sagittal, we turn the brain toward the side and we cut straight through and bisect the corpus callosum. We basically cut the 2 hemispheres in half with the sagittal cut. Now, I want to finish up by talking about brain injury and its effects. Your book is going to talk more about a bit more about this than I will, so make sure that you are reading up on this. So, traumatic brain injury is often going to involve a blow to a head. Now, your textbook is a little bit older, so I wanted to pull up some more recent stats. As of 2023, 2.87 million in the United States suffer from TBI each year. This can come from things like car accidents, falls, etc. Now, what are the current treatments for a traumatic brain injury? Generally, the goal is to limit further injury. We want to reduce swelling. We want to use medication to basically keep blood flowing to the brain. One of the things that I didn't really talk about here is that proper blood flow to the brain is going to be really critical for normal function of the nervous system. And areas that are very highly active require an increased blood flow. But notice that there's not really a focus on restoring function or preserving cells that are already damaged and dying. Now, why is it so hard for us to do more? Well, that's largely because of glial scars, or what your textbook refers to as gliosis. So what are glial scars? Basically, what's going to happen is that during injury, the astrocytes are going to basically swell near injury sites, and it's basically going to produce this scar tissue. This is also going to help separate healthy tissue from the damaged tissue. It's also going to protect and help restore the blood-brain barrier. Now, as you can see, the axon is damaged, but it can regenerate, particularly if we're talking about the peripheral nervous system. But they are ultimately going to be diverted from areas of damage. This axon is not going to be able to work through that glial scar. And that's because astrocytes and microglia release what is called an inhibitory extracellular matrix. These are basically chemical messengers that tell the regenerating axon buds where to grow. So you can see that it's basically going to try to stay away from that scar tissue. So different ways to treat it could be trying to inhibit this extracellular matrix. Another thing that your textbook talks about are things like neurotropic factors. Those may potentially also work, but they're very new in their infancy, and there's obviously more that we need to learn. So folks, that is pretty much it for today. Next time, I am going to be talking to you about neurotransmission, and we will finish up this first week's lectures. Bye, everybody.